Okay, good afternoon. Thanks everyone for joining us this afternoon. My name is Alison Mears and I'm co-director of the Healthy Materials Lab at Parsons. It's my pleasure to welcome you all to our fall lecture series, aspirationally titled A World Without Toxics. In this series, we'll begin to explore why and how we're exposed to toxic chemicals in our environments. We ask, how can we design healthier, toxic-free buildings to be part of a movement to build healthier buildings for all people? So a couple of housekeeping notes before we dive in. Please feel free to add any questions in the Q&A, not in the chat function, in the Q&A, and we'll try to get to as many of the questions as we can. Also, some good news for architects who are in the audience this afternoon. We're offering CEUs for this event, so look for the prompt in the chat to upload your information, and you'll see it in the chat towards the end of the presentation. So make sure you do that because I know it's October and everybody's scrambling to get their CEUs for the year. So our first presentation today is from Dr. Robert Wright from Mount Sinai's Institute of Exposomic Research. He will speak about the toxic chemical exposure that people may encounter in their everyday environments. The Mount Sinai Institute of Exposomic Research is developing a range of new techniques to measure environmental toxic exposure and its effects on human health. It has been our honor to partner with Dr. Wright and the team from Mount Sinai on a project to translate the outcomes of research into lead exposure in children to help, and in our case, it was to create accessible, accurate information for parents of research participants. So we have long been great admirers of Dr. Wright's research and the group at Mount Sinai. So we're really excited that he's able to join us today. A little bit of his bio. He is the Ethel H. Wise Chair of Environmental Medicine at Mount Sinai, and he has a transdisciplinary background in pediatrics, toxicology, genetics, and environmental epidemiology. He established the Mount Sinai Institute for Exposomic Research in 2017, and as a leading physician advocate of incorporating exposomics into precision medicine and public health. He directs the Mount Sinai Human Health Exposure Analysis Resource Targeted Lab Hub. His research seeks to link exposomics and phenomics with life stage, employing data science to address the complex problems in human health and disease research. A warmest welcome to Dr. Wright. Thank you for making the time to speak with us today.
Thank you, Allison. I'm just going to bring up my slides. Actually, I literally just submitted, I write a blog on Exposomics. I literally just submitted my October blog, and it was about language and how it impedes scientific communication, because all fields have their own language and they may refer to even the same thing using very different words. And I thought that was actually an appropriate blog actually for TASOC, because I obviously am not an architect and I don't understand architecture, but I think there's quite a bit I can learn about environmental health and the role that building and architecture plays in it. So I'm looking forward to the discussion. So I thought I would really just provide a primer of what Exposomics is for this talk, and then I'll take questions at the end. So what is Exposomics? There's various descriptions of what its definition is. It comes out of the world of environmental health. So initially it was really just about chemicals, but I think in the last few years, it's really expanded to include everything that is not genomics. So it includes things like allergy. This is a piece of pollen here on this field, mosquito-borne vectors or other types of what are called zoonoses or infections that come from animals. It can be things like viruses. This is actually the COVID virus. It's the social environment. It's things that you can't necessarily measure in the blood or urine, like light or noise. it's nutrition. It does include chemicals and it does include the physical environment, but it's actually much more than just the chemical or physical environment. And I think a more succinct way to describe what exposomics is, it's the study of all the health relevant environment we experience across the lifespan, which can seem very daunting, actually, when you think about it, considering how many years we've been around, and that going back in time to measure what happened to us during when our mother was pregnant with us could be very, very difficult. It could be decades ago, but there has actually been a number of advances that actually make exposomics, I think, much more feasible than people realize. It's much more advanced, as I said, than you may know. So it is very different than a lot of other sciences. Genomics has really dominated the scientific world over the last 20 years, even 30 years, especially since the completion of the Human Genome Project around 1999. Genomics really is, in a way, much simpler than exposomics. For one, your DNA sequence is the same in every single tissue in your body, in every single cell in your body, and it doesn't change over time. So you only have to measure it once, and it should be the same when you're 10 years old, or as is when you're 30 or 60 or 80 years old. So in that way, even though it's 4 billion base pairs, it's actually a much easier measurement than the exposome, because the exposome does change over time. And the exposome isn't just one type of test. So with genomics, we do what's called DNA sequencing, where we just sequence the base pairs, whether they're adenine or thymine or cytosine or guanine. There's just 4 possible outcomes, and we measure them 4 billion times, which is a large amount of information. But exposomics isn't just one type of test. It's actually many, many types of data. So it will include questionnaires. So knowing where you live, knowing what your occupation is, and particularly if you want to go in the past, knowing if you've smoked, all those sorts of information, all that sort of information, levels of perceived stress or perceived racism, all those things actually are typically collected by questionnaires. It also uses a lot of geospatial modeling. So we measure air pollution, we measure green space, we measure noise. So some of our faculty actually work with NASA. And we actually use NASA satellites and satellite remote sensing to actually estimate different climate variables or air pollution variables. I think there's a lot of work being done in using pictures. So a lot of times we'll now ask people to take a picture of all the cleaning products in their home, and that gives us some information on what they're exposed to through those cleaning products. People are trying it with food as well. My daughter actually takes pictures of what she's eaten and posts it on Instagram, and there are people who are working on whether or not we can get nutritional information from photographs. And of course, there's always going to be biomarkers. Sometimes we use blood, sometimes we use urine. Some people use things like exhaled breath. So there's all different types of matrices, so to speak, in which we measure different environmental factors that we have. There's also mobile devices. So wearable devices, many of you probably have a Garmin or an Apple Watch. Almost everybody on the planet has a cell phone these days. And these devices exchange data with the internet, and they can tell us where you are. Which can be very, very useful, particularly for GIS measures. So we can actually get a more accurate measurement of, say, climate or local climate or air pollution, but also your response to the environment. So we have a sense of your heart rate and your breathing rate, oftentimes from these wearable devices. And all this information, in theory, can be linked to other types of omics, such as epigenomics and transcriptomics and proteomics, which are other sort of molecular biology based assays. So exposomics is different is because it doesn't really have that sort of molecular biology. It's not related directly to DNA. It's also different because what you get exposed to actually is dependent on your age, but it's also dependent on the year at which you're a particular age. So a good example was lead poisoning. So in the 1960s, the average blood lead in the United States, all comers, was about 7 or 8 micrograms per deciliter. It got taken out of gasoline, it got taken out of lead paint, and it reduced the average blood lead to what it is today is less than 1 microgram per deciliter. However, if you look at people of different ages, you'll find that adults, it's even lower than 1 microgram per deciliter. And it's actually highest in children who are about 2 years of age. And that's because when you're 2 years of age, there's sort of a normal hand-to-mouth behavior that occurs. And so, and 2-year-old toddlers are crawling around the floor, or they're touching windowsills, and they're putting their hands in their mouth, so they're ingesting lots of dust. So whatever in their dust in their home can actually get inside their bodies. So 2-year-olds are actually the age at which we see lead poisoning. And then because behavior changes very rapidly across childhood, you won't see lead poisoning in a 10-year-old. It's very, very unusual. It's almost always in a very small child. Now, if you do see something in a 10-year-old, then we usually have some unusual source that actually may be causing the lead poisoning, but it's unlikely to be the home, so to speak. So there's all kinds of different ways that we can actually predict the environment. It's different at age 2 than at age 10. It's different when you're 20 years old. It's different when you're 80 years old, whether what the types of foods that you eat differ, and the places that you live will probably differ, and the types of housing that you live in, where you work, well, all of that will differ based on the age and period effects. So what happened in 1940s, the types of buildings that were built, and what is occurring today are very, very different. So there's this period effect as well as an age effect. And that makes it also very different than genomics, because genomics doesn't change over time. How do we know exposomics is important? Well, there's a lot of data that show that zip code is a better predictor of your health than your genetic code. In fact, mortality is better predicted by zip code. Zip code is a measure of place, where you are, where you live. It correlates probably closer to where you work than your genetic code. And it's a much better predictor of your health. It's a very crude measure of place. If you think about how many thousands, potentially millions of people live in your zip code, it really isn't telling you all that much about place, but that small amount that it's telling you actually predicts your health better than your DNA. And this is probably because zip code is a proxy for other types of exposomic measurements that vary by place, things like socioeconomic status. So zip code actually is a proxy for that. It's also a proxy for things like racism. So think about redlining. Redlining was a practice by which people of color were segregated into particular neighborhoods, and those neighborhoods often had more environmental pollution. They often had lower property values. They often had more discrimination in those areas, particularly in the housing and other financial issues. So zip code is actually a proxy for that. It's also a proxy for environmental pollution, because there's actually more air pollution in Harlem than there is on the Upper East Side. And that's in part because the bus depots in the city of New York are actually congregated in Harlem rather than evenly distributed across the city, even though that probably would have been more financially manageable. They decided to put all the bus depots that idle in the morning in Harlem. So, and race is not a social, or excuse me, it is a social construct. It's not a biological construct. And race itself, when we do studies, when we're adjusting for race, we're not adjusting for anything genetic. We're adjusting for racism and all those correlates of racism that I just talked about. The same is also true for SES. They are proxies for higher pollution, poor access to healthy food, poor educational opportunities, and higher levels of stress. And that's why zip code is a much better predictor of health than your genetic code. And where you are actually is a very good predictor of your exposome. And when I say exposome, I'm talking about the social exposome, the physical, the chemical, nutritional exposome, all aspects of it. And why exposomics, I think, is exciting, because it's about measuring those factors that actually are causing the health impact, rather than using things like zip code or race or SES, socioeconomic status, as a proxy for those factors. We're actually trying to delve in and actually figure out what they are so that we can actually modify them and intervene. So that's why exposomics is important. I've also talked about what exposomics is. So how do we measure the exposome? Well, as I alluded to this several different ways. One way is to measure it in an assay. So these are blood tests or urine tests. on very, very expensive instruments. We can measure nutritional factors. We can measure chemicals. We can measure metabolites from our food and our diet. We can measure hormones, which arise, ebb and flow based on our response to the exposome. And then there's two types of tests that are done, or what we call targeted tests. These are quantitative, so we know the exact amount, say lead in the blood. And there's untargeted, where we do a scan to try to find everything that's inside a blood or urine sample, but it's not really quantitative. It's at best semi-quantitative. So we know the relative relationship to each other, but we can't put a number on how much somebody's actually exposed to. But we know more information about how many things you're exposed to. And I'll talk a little bit about teeth and hair in an upcoming slide, but there are some ways to actually go back in time. And so Allison talked about Dr. Aurora and the tooth biomarker, I believe earlier, and there's some information that actually can be gleaned from things like teeth or biological tissues like teeth that tell you the timing of past exposure. And then there's what I often refer to as the external exposome. Rather than assays, these are models. So we typically mine public data sets. They may be NASA data sets that give us information on air pollution or weather from satellites, or they may be monitors that a city has placed for light or noise, or other types of monitors and wearable devices that are crowdsourced, like for stress and different types of hormones, perhaps. But they're not technically inside the body, but they're experienced. And then there's the social exposome. These are I talked about briefly in a previous slide, things like racism, stress, but also positive things like coping and resilience. We can also mine public databases for exposure to violence on the community level. And we can also measure things like social supports, all of which have profound impacts on our health. And these can either be measured from databases or from questionnaires, but everything on this slide can be mapped. In fact, I'm a big fan of mapping everything. We can map assays. We can map the external exposome by definition as a map, but we can also measure the social exposome and map it. And by mapping it, we start to see patterns of where things are occurring, and where things are occurring give us information on why there may be more or less greater health effects in a particular geographic area. So the internal exposome, the assays look kind of like this. This is what's called a chromatogram. Chromatograms, actually all of these peaks in a chromatogram represent a chemical. And there's probably at least 100,000 chemicals in your blood that we can measure. We don't know them at a quantitative level. We can't tell you what the concentration is, but we can identify at least 100,000 of them. We don't have good databases to tell you the name of the chemical. But what's interesting about different research approaches, if the peak is reproducible, so we know it's a chemical, and we know it's there in many, many people, we can actually look at whether or not that peak is related to a disease, or it's more common in a diseased person versus a person who doesn't have a disease. And we can get a better sense of whether or not that chemical is biologically active. And then we can do the chemistry to actually identify that chemical. So a lot of this is because it's a relatively new field. We don't have the databases that genomics has in order to identify DNA variation yet in exposomics, but that's being worked on. And every year, those data sets get better and better, but there's still a long way to go compared to genomics. But these assays measure thousands of chemicals in our bodies, and these chemicals are oftentimes nutrients or metabolites. They're consumer products. They're things that came into our body through the air and the water and the food that we eat. And then these are targeted assays. So these are assays that we quantitate. It's a slightly different approach than the untargeted assay. And we can measure hundreds of them. And they include things like plastics. Phthalates are soft plasticizers that are found in different types of soft plastics, and also things like soaps. Phenols are similar in that regard, that they're also found in things like soaps and other types of consumer products. There are dyes and additives to food and beverages. There are things that people smoke, drugs of abuse, but also cigarettes or even vape. There's actually ways to measure chemicals that come from vape and chemicals that come from smokes. We can actually distinguish whether somebody's smoking versus vaping. We can measure VOC, volatile organic chemicals. Some of these are coming off things like carpets. That's that sort of organic smell that you get from certain products. And the ventilation in a room can actually determine the level of exposure. So it's not just that a carpet, say, is off-gassing a volatile organic compound, it's also whether or not that room is ventilated enough to take that compound out at a rapid rate. And then there's all kinds of other things that we measure as well, such as nutrition, response to environmental factors, such what's called oxidative stress, which is a form of inflammation, is what most of these biological response assays actually refer to. You've probably read about perfluoroalkyls or polyfluorinated carbon compounds. These are PFAS and PFOA. These are sometimes referred to as forever compounds. They're found in nonstick cookware and a whole bunch of consumer products, and they have half-lives, meaning that the length of time that they stay inside the body is measured in years. So everybody on this call, if you gave us a sample of blood, you would have a measurable amount of these perfluoroalkyl chemicals, these forever chemicals in your body, because they're so pervasive and they have such a long half-life that everybody I would predict would have a measurable amount. And there's a lot of research being done on their health impact. And then I do a lot of work with trace elements, things like lead, cobalt, cadmium, manganese, other trace elements. We also can measure them in our laboratory at Mount Sinai. And then I alluded to this earlier. So one of our faculty, Dr. Manish Arora, is a dentist and a chemist, and he's developed an assay that actually measures chemicals inside teeth. Now, it turns out teeth grow a lot like trees, and so they form rings, or growth rings, just like the rings of a tree. And these rings are formed on a daily basis, starting during pregnancy, and using very sophisticated techniques such as laser ablation and very sophisticated instruments for measuring chemicals. ICP-MS is inductively coupled plasma mass spectrometry. It measures trace elements. He can actually measure the amount of a chemical inside these rings. And there's a particular ring that's formed on the day you're born called the neonatal line. It's pointed out in this graph. It's referred to as the birth line, but that's the ring that's formed on the day that you're born. It histologically looks different than all the other rings. So once he identifies that ring, he actually has a calendar date, and the rings formed on one side are the rings that were formed during pregnancy, and the rings formed on the other side are the rings formed after pregnancy, and each ring represents one day. Obviously, these rings are very, very tiny. These rings are formed throughout your life. That's why your teeth will tend to crowd as you get older, particularly if you haven't lost any teeth. So your teeth actually never really stop growing. And this technique works really well in children because children lose their baby teeth, and then parents can give the tooth to Dr. Arora, and Dr. Arora can do analysis of past chemical exposures. In adults, you would have to have your tooth pulled for him to do it. And then hair also has some growth patterns. So hair obviously doesn't grow as slowly as teeth, but you can get some information on past exposures to chemicals using hair. Because hair has growth patterns and growth rings as well. And then there are all kinds of things that we can measure using GIS or geographical information systems for what I've referred to as the experimental exposome. These are just some examples of the commonly measured things. So there's a lot of interest, obviously, in climate change and how climate change impacts health. So some of our faculty are working on temperature and climate and health. And so this is a map of New York City and average temperatures in 2011. We have maps much more recent than this one, actually on a finer scale. So at the time this map was done, we were measuring average temperatures in a 1 kilometer grid. Now we have 30-meter grids. So the technology is getting better and better and better so that we actually have a better resolution of what the average temperature is in a particular location. We can also map whether or not you're exposed to green space. There's a lot of interest in the role of green space in promoting positive health. We're also measuring daily air pollution. So this is a map of New York and the Philadelphia area in terms of levels of air pollution. You can see a gradient there with the darker colors representing the highest levels of air pollution. We make maps for traffic patterns and noise. traffic and noise are very highly correlated to each other. We look at the built environment, walkability scales, but also access to healthy foods. And I think one of the things that we're starting to do that I think is very exciting is looking at mapping social media content. So this is actually a study done by one of our faculty. Twitter actually has geospatial information in it every time you post something on Twitter, and then using a process called natural language processing, which is a computer algorithm to find certain types of words. He actually searched for mean tweets and then plotted their geospatial distribution in the Boston area. So this is where all the meanest tweets were actually coming from in Boston. And I believe the year was 2014. And then the other piece of the puzzle for Exposomics are wearable devices. So as I said, everybody on this call probably carries a cell phone. A cell phone is a type of wearable device, and then it has GPS information, and it's part of the Internet of Things. These are apps and that can be found in your phone, in a car, or appliances that exchange data with the internet, which tells us something about the amount of noise that you're exposed to, the amount of light that you're exposed to, and other types of variables. The GPS data can give us a finer resolution on the amount of air pollution exposed to or the climate that you're exposed to. But also, as I said, because it's measuring things like your physiologic data, like your heart rate, and your respiratory rate, we also get a sense of your response to all these different environmental factors. And when it's combined with remote sensing, we get a much more precise exposure measurement. And we can also combine it with assays. So if we have one of those untargeted assays and we know geospatially where you've been in the previous 24 hours, we actually have a better sense of where some of these exposures may be coming from. So all of these things are interrelated. And in fact, I believe everything can and should be mapped. So this is a map of childhood lead poisoning in New York City. These are housing violations, and they correspond highly correlated with about lead levels in children. And it tells you something about where the hotspots are, where the environmental issues are likely to be. Obviously here in northern Harlem, northern Manhattan, but also in the Bronx are two of the areas in which we see the most lead poisoning, but also certain parts of Brooklyn. Not all of Brooklyn, but certain parts of Brooklyn have a lot of lead poisoning. So these are areas in which interventions should be targeted. So maps are very, very informative for Exposomics, and I think they're the key actually to Exposomics, and I believe everything should be mapped, whether it's air pollution or blood lead data, or even those untargeted assays that I talked about earlier. Wearable devices, as I said, there is actually an Exposomic wearable device. It's actually very simple. It's a silicone wristband. You wear it for a certain number of days, and we take it off, and then we measure what was in it. So it tells you what was in your external environment. It's not obviously internal, but if we combine it with a GPS device like a Fitbit or a cell phone, we do have a sense of what the environment is in the areas that you've been during the time you're wearing the wristband. So there's a lot of ways that we actually combine these different types of measurements. And I think Exposomics is really going to complete what I call the complex disease puzzle. Complex diseases are diseases that are very, very common. They have both genetic and environmental causes. The genomic causes or genetic causes of them have been dominated the research world for the last 20 years, but there isn't much you can do about your genetics, like whether or not you're predisposed to a heart attack or Alzheimer's disease. There's no gene therapies for that because it's not caused by a single gene. There's several genes typically involved. And there's also many, many environmental factors. What I think Exposomics, why Exposomics is important is because you can modify your lifestyle, you can modify your environment. And so I think it is a way in which we can actually reduce our risk going forward. And I think Exposomics is actually going to be probably a dominant research field going forward as we start to understand the role of the environment. Also, even though we completed the human genome about 20 years ago, as a physician, I can tell you medicine has changed only incrementally in that amount of time. And that's because genomics by itself doesn't explain very much about why these complex diseases are getting more and more common, and hasn't led to a lot of interventions or treatments that work. I think exposomics, when it's particularly even when it's combined with genomics, is going to be much more informative and much more likely to actually give us a better understanding of why these diseases have become so common, and hopefully intervene. So I'm going to spend the last few slides telling you a little bit of what we do at Mount Sinai at the Institute for Exposomic Research and what's happening here. So we have an annual symposium on exposomics. We call it the New York City Exposome Symposium. The most recent one was in July of this year. It was an in-person hybrid. It'd been Zoom only in 2021. But this year we thought things got a little bit better. So we went to hybrid, and our keynote speaker was the director of the National Institute of Environmental Health Sciences, which is one of the institutes of NIH. We have a partnership with the University of Brescia in Italy. So we actually do a second Exposome Symposium there, and that's scheduled for April of next year. And we have collaborations with Ben Gurion University in Israel and the National Institute of Public Health in Mexico. So we're also planning to do some symposiums there in the next 1 or 2 years. We also have an annual clinical climate change conference. This year was themed on mental health and well-being for patients and clinicians. And our keynote speaker was Admiral Rachel Levine. She's a physician. She also is a Mount Sinai alumnus, and she's the Assistant Secretary for Human Health in the Department of Health and Human Services. And she gave a keynote on mental health and climate change. We have a P30 Core Center grant from NIH, which does pilot grants, among other things. It supports our laboratory, it supports some of our data science initiatives and measurements of health. But it also has a community engagement core, which does a lot of work with community advocacy groups and healthcare professionals to try to understand what are the important issues in the local community, particularly in Harlem and the South Bronx, so that we can actually help folks in partnership to reduce their toxic environmental exposure and promote a more healthy exposome, but also deal with issues of environmental justice. Our CTSA grant recently got funded. It includes a module called Exposomics Precision Medicine. So we're working to collect addresses on folks at Mount Sinai so that we can actually link them to all those maps that we talked about earlier and get a better sense of what their environmental exposures are, including things like crime and violence, green space, air pollution, traffic, and industrial air pollution. And what we call a geomarker database. So this is actually in its early stages, but I think it's an exciting new area that we hope to link to our precision medicine initiatives at Mount Sinai. And we're also part of an NIH program called HERE, the Human Health Exposure Analysis Resource. So our laboratory does both untargeted chemical assays and targeted chemical assays I described earlier for NIH-funded investigators doing research. We do the tooth biomarker that Dr. Aurora talked about, or Dr. Aurora does, as well as part of that program. So we're very, very integrated with the NIH programs in exposomics. And that is my last slide. I'm happy to take any questions.
Amazing. Thank you. That was extraordinary, very fast. I hope everybody got all of that. We're going to have a quiz now, right, Bob, and just see what people took away. I had a question. I'm going to kind of build from Grant's question. Thanks, Grant. About particularly as we think about designers and architects, do you know, many of, particularly the chromatograph, how you can detect that whole 100,000 different chemicals in the human body. And as we think about the peaks and troughs there, thinking about which of those, particularly the synthetic chemicals that we have in our bodies, thinking about where they come from in our interior environments. So if we're thinking about our audience, architects and designers, you know, how can we do a better job about not bringing those kind of chemicals into our interior environments? So the question for you, I think, would be like, what are the worst offenders in your research in terms of those kinds of chemicals? What should we be targeting as our number one and number two chemical to try and eliminate them from our environments, our built environments?
I think a very simple one is carbon dioxide, particularly in the workplace. You know, I think proper ventilation in rooms is really, really important and can reduce that. I think we've all sat in conference rooms that were poorly ventilated and got sleepy about halfway through. It's not always just the speaker. Some of it is the CO2 levels go up. One way to find that out is there are relatively inexpensive CO2 monitors that you can place in rooms, and you can actually see how well the room is ventilated, not just when it's empty, but also when it's full, and see the change over time. And I think that would give you some interesting information on, you know, the types of rooms that tend to get you know, higher levels, and there is a health impact to that. So I do think that, you know, that's an easy thing to do. Also, if your room is well ventilated, any type of volatile organic chemical that's being, you know, off-gassed from some product, it's going to be in lower concentration. So I think focusing on ventilation, on good ventilation, is actually very, very key for a lot of issues. And I think would probably be where I would recommend efforts be put, because it's going to help almost no matter what the chemical is. Obviously, you don't want to introduce toxic chemicals if you can avoid it, but if you have good ventilation, you're at least going to mitigate some of their impact.
Yeah, and back to kind of Grant's question too, I think, you know, he speaks to the complexity of those chemicals that are available with our environments. If we think about that, both the airborne and the particulate chemicals that are, the particular chemicals that are falling down, as Maida Galvez says, into the toxic soup that is a carpet, for example, or airborne into the air, just the interrelationship, the interaction of those chemicals in the environment, both in the environment and then as you absorb them into your body, you know, through those different pathways. You know, it feels to me like, you know, part of that complexity is being tackled by your work. And if you could talk about that a little bit.
Although I will confess that indoor air is a big black box in the environmental health world. So all those models I showed are primarily for outdoor air. There's a lot of outdoor air monitors, and we're very good at measuring and modeling the outdoor air. Indoor air does not correlate with outdoor air. So we know that they're independent. Indoor air is harder to measure because it requires, you know, the person or the owner of the building allowing you to do it. I think there's a lot of research need, both in measuring and quantifying the levels of particulate matter and different chemicals in indoor air, but also understanding what are the sources of those chemicals, so that perhaps we could prevent their exposure, but also model them so we don't have to measure it in each new building. If we know sort of the characteristics of the building, maybe we could predict that, oh, this building is going to have a lot of these VOCs in it because of the type of carpet that's in it and the type of windows. And it's, you know, the ventilation is, the CO2 level is probably a proxy for ventilations, and the CO2 levels tend to go up, which suggests that, you know, they're probably also not just breathing CO2, but also breathing these volatile organic chemicals because it's not well ventilated. That sort of work still needs to be done. It is something that people in the environmental health world will tell you, if they're honest, that we don't know a lot about indoor air, what's in it.
It feels to me that that's one of our jobs then, is to try and, you know, we know there is a correlation between some of these chemicals and human health, and possibly many of these chemicals in human health. And so if we have a decision, for example, about, you know, a high VOC product, whether it's a carpet or it's a paint, then that move to low VOC or no VOC seems to be like just as a rule of thumb, a better thing to be doing. As we kind of, as the research catches up with practice, for us, I think we would be advocating, obviously, for healthier materials that didn't have some of those synthetic chemicals, particularly the PFOSs, PFOAs, and the other, you know, the very long-term chemicals that are going to be in our body for a while, so it feels like that would be good practice to do. I know there's a question here about monitoring interior environments, and there are, as we've seen, a number of new small kind of residential devices that will measure the particulate matter in interiors, and also I think the CO2 levels as well. Have you had any experience with those? It feels like you're in the monitoring kind of space.
A few. I really shouldn't advocate for one product over
No.
another, but they do vary in the quality of the data. So you should look into what's known about how well it's calibrated and how well it's been validated before purchasing it. They're not all the same, but some of them are quite good. And many of them, even if they're off in the amount of particulate matter, say, or whatever it is they're measuring, the pattern of the ebb and flow across the day will be accurate. So so sometimes, I think sometimes our, say, industrial hygienists dismiss the monitor, but if it's off by a known amount and it's measuring the ebb and flow, it's giving you some useful information. So it's not noise, in other words. But yes, I think I'm a big advocate for us collecting more and more data about indoor air, because most of us spend more time indoors than outdoors. So it is not only a big black box in terms of what we're breathing, but it is actually probably the more important of the two measurements. We spend much less time outdoors, most of us, than indoors.
Yes, yeah. I mean, there is kind of the statistic that we quote that we spend, at least 90% of our time indoors, and so that environment's kind of critically important for us. There is, thank you, Rebecca, for your question with regard to ventilation in healthcare environments. Do you think we'll ever return to operable windows? That seems to be a move that the Europeans are progressing towards. I mean, in our research, we often say that the interior air is much more toxic than exterior air, and I think that's why the Europeans are looking to open windows. It depends, I guess.
It depends on where you are. If you're in Los Angeles, that might not be true, but to be honest, if you're in New York City, that might not be true. But if you're in a suburb, it probably is true. If you're in a rural area, the outdoor air probably is cleaner. And also will vary by day. So, certain days are just going to be bad days in New York, and other days are going to be good. But having the option of opening the windows, I would be a big advocate for, because I do think, there's the ventilation is a big part of why sick building syndrome occurs, and having good ventilation is really important for health.
I know that there are, Duncan's talking about ASHRAE guidelines for minimum ventilation standards. So part of those, I think, are around the fresh air issues that you talked about before, Bob. I'm not sure that they're necessarily setting guidelines for toxics in that air, Duncan, but that's maybe a question that we could get back to you about. I did have a question for you, Bob, about stress. You did mention stress as one of the things that you measure, and stress being connected to environments. Could you talk a little bit about the impact of stress on people generally as a kind of social, as part of that social exposome that you talked about? What does stress do to people? What are the negative impacts of stress?
Well, yeah, stress is complicated. It's not always bad to be stressed. Being resilient in the face of stress actually can have a lot of positive benefits. I think the way stress gets into the body in a more toxic way is when it's chronic, or when it's very, very severe, like a PTSD type of situation. But stress is complicated because it's really your reaction to stress that matters. So we talk a lot about stress, but the reason why there's so much variability is that different people have different reactions to it. And there's all kinds of reasons why people have an unhealthy reaction versus a healthy reaction. A lot of it has to do with things like social networks and coping skills and behaviors that were probably learned in childhood. There's certainly a genetic issue as well, or predisposition to things like PTSD. But a lot of it actually is probably, much more of it is probably things like learned behaviors or social networks, which we don't typically measure very well as researchers. So stress is very, very complicated. It certainly is higher in poorer communities and than it is in wealthier communities, but it's not absent in wealthier communities either. And nor is it universal that people who live in poorer communities have adverse reactions to stress. Some of them are resilient. And so what we're trying to do is understand what those resilience factors are, and perhaps even advocate for them as potential prevention or intervention tools for communities that live in areas of high stress. And housing stress is a major stress, as well as financial stress, and they're usually intertwined, actually.
When you say housing stress, you mean lack of access to housing?
Lack of access to housing, or financial stress related to housing, or living in a house that has, lead-based paint, is broken down, has poor water quality, is in an area of high air pollution. All those things, lack of access to air conditioning during a heat wave, all those things at some level are housing issues, and they are contributors to stress as well.
And could you speak a little bit about that idea of intergenerational stress, that you're inherit in some communities, that stress is something that is inherited generation to generation?
It is something that there's certainly a lot of interest in it. It's very difficult to tease out the cause. Poverty is, in a sense, inherited, just like wealth is inherited. So how much of it is intergenerational in a sense of being transmitted, or intergenerational because social economic status tends to correlate across generations? I tend to think more of it is actually due to that things like socioeconomic status typically don't change from one generation to the next. There is some evidence, though, that some degree of stress is biologically transmitted from a mother to a child through hormones and other things that change marks on DNA. So I think there is some evidence that occurs. But I think the vast majority of intergenerational stress is actually what you're seeing is things like racism and poverty that simply don't go away across generations.
I mean, yeah, the again, for us, as architects and designers, really thinking about the importance of, safe, healthy, affordable housing as being a real imperative.
Yeah. And walkability. It's paradoxical that I think it was in the 70s and 80s that neighborhoods started being built without sidewalks, which really, makes it harder to go for a walk if you have to walk in the street. And you tend, particularly for children, you tend to tell your kids not to go into the street because you don't want them obviously hit by a car, but there's no sidewalk, it kind of inhibits them from happening. So that's something that I think, walkability is something that I think would be a big help, that architects, thought about those sorts of issues so that there are places for people to get simple exercise. Just walking is a great exercise for your health. And we can design neighborhoods to promote walking rather than to inhibit walking.
There's a question here from Bryce. Thank you, Bryce. And he's asking whether there's any studies being done which chart the year of a building's construction with the types of exposure to residents. So looking at some construction that happened in the 1950s, for example, and maybe, something that then post-lead, well, the elimination of lead, at least and then a modern building, any of those kind of longitudinal studies on different environments.
Epidemiologists have not focused so much on buildings as they have on people, but I think it's a great idea. So to look at buildings and assess the health impact of a building on the people that live there and how that impact changes over time. I think that's a great idea. It's not a typical way a epidemiologic study is designed. It's usually you follow people, and then they may move to different buildings, and you might assess them when they're living in different buildings. But if the unit of analysis is a building, I've not seen that actually. I think it's a great idea. It would be difficult to do going backwards, because you'd have to track down everybody who used to live in a building, but it is something that could be done going forward in time.
There's an interesting question here about the gender disparities, particularly things like women's breast cancer, for example. Have you focused at all in that area, gender disparities between people?
It's not, I'm a pediatrician, so I primarily look at children's environmental health. I'm not an expert in breast cancer. But there are some faculty, Susan Teitelbaum is one that actually have done some research in breast cancer. But there's a lot of press about the genetics of breast cancer, but what often gets lost is it's actually a minority of the breast cancer cases are due to genetics. Most of it is likely due to environmental factors, many of which haven't been discovered yet, although smoking takes up a pretty good size of the environmental piece for breast cancer.
I know that somebody is asking about the Exposomic wristband. Is that something that they could contact the center about, or is that something that's generally you could Google and get one?
You can Google and get one. There's a company called My Exposome. A little pricey, but they'll send you the wristband. You can wear it, and you can send it to them, and they'll analyze it and give you the results. Our laboratory does it primarily for research, so we don't do it for personal data. We're not a CLIA certified laboratory, so we really can't do it individual level, but there is a company that will do it.
Do they then share that information with researchers like you? Do they collect information like that or no?
The company, not to my knowledge. No, I think they're in it to make money. So, but they will do it for you, and it's a good laboratory. I know the person who runs it. She's an excellent scientist, and I would trust her results. I think the problem is, a lot of chemicals, we're not really clear what they do. So we can tell you what you're exposed to, and in many cases, we can tell you what the sources for that exposure likely are. But like 23andMe and Ancestry.com, it's not telling you with any certainty that you're going to get a disease going forward, but it will give you information on what's in your environment. And some of those chemicals likely are going to be toxic, and at least gives you the information. I always say is power. So if you have the information, you can decide whether you want to reduce, or how much risk you're willing to take, and whether you want to reduce that risk. And in that regard, the bracelet is better than 23andMe, because there's really nothing you can do about your genomics. It is what it is, and it's not going to change. But your environment, through behavioral modifications or other types of interventions, just switching products in some cases, you can actually change your environment. So I do think in that regard, it's much more powerful.
Could you talk, Dr. Shana Swan, we had her speak last year, I think, guys, a little bit about her work. I know there's some, questions about plastics, a lot of questions about plastics. Could you talk a little bit about her work? Is that something you would talk about?
I think she studies reproductive health,
yes.
Which is a little different than what I do. I study children's health. And I know she wrote a book about the impact of different environmental chemicals on, I think, sperm motility.
Yes.
Other than that, I'm not that familiar, to be honest, with her work. It is a little bit outside my field, which is children's environmental health and child development.
Yeah, if you're interested in her work, it's really the connection between phthalates and bisphenol A, the impact of those plastics basically on men and women's reproductive health. And her book is called Countdown. So that's something to look out for, and we can share that afterwards too, or somebody can put it in the chat. Ava, if you could put it in the chat, that would be really great.
There's a lot of interest in plastics in general, obviously because they don't degrade. There are there have actually been some recent studies that show nanoparticles of plastics are measurable in our blood. So plastics are so ubiquitous, and they do break down into nanoparticles, that the particles themselves, regardless of what they're composed of, can actually be measured in blood and urine.
And that's something that is ubiquitous through ages, from a fetus all the way through to end of life. We're all exposed to this, right?
In all likelihood, there's only been it's only a recent finding. The laboratory technology to do that kind of assay is relatively recent. I would expect it to be, but I think I would be overstepping what's known if I said it was. But the fact that a relatively small study could measure nanoparticles of plastics in everybody they measured in is a little worrisome, that it is pretty ubiquitous. It was a study out of the Netherlands.
Burgess, do you see any questions there that you'd like to point out? Burgess is also helping me review questions here. We've got lots of questions. Thank you, everyone, for such thoughtful questions.
Yeah, I like this question from Lisa. I'm not sure if you'll be able to answer it, but Lisa asks, is there comparative data showing how these metrics have significantly changed in the last 5, 10, or 20 years? Any particular chemicals that are the greatest offenders and what they are attributed to? So I think you maybe have answered a bit of the second part, but I think the change over time is interesting.
Sure. Well, exposomics is still a pretty young field, so we don't have a lot of data on thousands of chemicals. Now, NHANES, which is the National Health and Nutrition Examination Survey, is a study that's run by the CDC. And every 2 years, they measure approximately 5,000 adults in the United States, and they are designed to be representative of the US population. And they've been measuring a core set of about 200 chemicals since roughly the late 1980s till today, every couple of years. And you will see trends with certain chemicals going down and up. Now, some chemicals like PFAS weren't measured back in the 1980s or more recent, but we are seeing trends of them going up. If you look at the NHANES data, lead has actually decreased. In the average blood lead of a child in the late 1980s was probably about 5 micrograms per deciliter. Now it's less than 1 microgram per deciliter. So you will see these patterns where some things go up and some things go down. Some of it is predictable based on the types of products used. Others are, it was interesting when some of the research came out that suggested BPA is toxic. BPA levels dropped without any kind of government regulation. However, one of the concerns that the field has is what typically happens is if a chemical such as BPA gets some bad press, then the industry will switch to a different chemical, which is very similar. So BPS, which is very similar to BPA, and BPF are now used in plastics, and they're both bisphenols. They're just a different type of bisphenol, just slightly different with their chemical structure, and they don't get tested for. So whether or not they're really safer is the question. They're definitely different, and we start to see those chemicals rise in NHANES as BPA fell, and that just suggested that industry was just switching from one chemical to another. They're sometimes referred to as regrettable replacements because they may actually be as toxic, in some cases possibly even more toxic than the chemical they were replacing.
Yeah, hugely problematic, a hugely problematic problem. I think we have time for just one more question. Elise has a question, I think, that is something that we are obviously also very invested in. Is, has your research been able to impact policy or regulations on materials acceptable to be produced and consumed? If not yet, is that a goal for the future, particularly as we think about children?
Well, I'll be honest, I have what many people think of as a cynical opinion about regulation. TSCA, which is a Toxic Regulation Act, which was passed in the 1970s and was recently amended and passed again under the Trump administration, has banned 4 chemicals
Mhm.
in total.
Mhm.
I don't think regulation works. I think industry finds ways around it. If you read the legislation, it sounds fantastic. It sounds like it's going to protect people and keep everybody safe. But the reality is there's all kinds of loopholes that someone who's not a lawyer would never see. And I actually think the best way to regulate is information. I'm a big fan of apps that, people will I think one day people will have an app and you'll buy a product and the product may not tell you everything that's in it. There'll be a database from a company that has measured what's in it using those untargeted assays. And that app will be able to tell you what's in it, and you as and what the potential health impact is, and you as a consumer will be able to compare app A or product A and product B, perhaps even inside the supermarket, and you can make your decision. And I think that kind of information, which hits companies in the pocketbook, will actually make them think about whether or not their product has something in it that may be toxic, and actually try to avoid those chemicals. I think that's going to be far more effective than legislation. I hate to say this, but I just think legislation doesn't work. But I think information will.
Yeah, I think that's right. And bottom line is, if you don't know what's in something, if you're an architect or a designer and your manufacturer, your product supplier isn't giving you the information you need about the ingredient content of a product you'd like to install, anywhere, then don't use it. That's really a bottom line for us, is we need that kind of transparency. We need to know what we're doing so that we can do a better job and we can eliminate some of these really bad, the forever chemicals and the rest of the list there from the products we use. So we're as much a part of the solution as anything, I think, which is a great opportunity. It gives us a little bit of power there. So thank you very much, Bob, for your great presentation. I'd just like to, and thank you for making the time to be with us. Really informative, very helpful for us to.
And I'm happy to share my slides if anyone wants to see them. I saw that someone asked for the link for the climate change. That should be in the slides, so I'll share those. Absolutely.
And I just had one final thing. This is Bob's presentation was the first in this series, so look out for another presentation in 2 weeks on October 20th at 12 noon. That'll be included with all the information that we send out to you probably tomorrow or the next day when Ava gets to it. And so we'll continue this kind of journey to try and remove these toxic chemicals from our built environments. Thank you all for joining us today. Great conversation. Thanks for Bob for that great presentation. Thanks very much, everyone. Bye-bye.
Bye-bye.